So all of these functional groups have only two choices, and here's the two choices on the menu here. Well, since we're at a low pH, we want the protonated form, which happens to be neutral in this case. Notice that the protonated form is not always neutral. The protonated form of an amine has a positive charge, but it just so happens that the protonated form for tyrosine is neutral, so you have to stop and think about it. So at this pH, what's the net charge?
0.
Yeah, we'd add the +1 and the -1 to get this 0 over here. Okay, good. So this is the type of question you're pretty sure to see on the test. He's going to say something like, draw tyrosine at a pH of 5. And then what do you have to be able to do? Well, first of all, you have to be able to find the correct side chain from the table and draw it correctly. But then you also have to be able to figure out what the correct forms of the main chain nitrogen, main chain carboxy group, and side chain are going to be. And then he might also ask you what's the net charge at that pH. In fact, he probably will, and this is the way we work that out, painstakingly, step by step, by going through all of this. We're about out of time, but why don't we do another example like this? So I guess we'll stick with the tyrosine. Let's suppose that we had a pH of 9.6. Let's draw what tyrosine would look like at a pH of 9.6. And let's figure out what its net charge would be. So let's think about that step by step. So we start with this functional group. For this functional group, is the pH high or low?
pH is high.
Because it's higher than 2.2.
Right.
So it should be deprotonated.
Right.
And we know that gives us a negative charge. So far, so good. Now, how about for this functional group? Is the pH high or low?
It is low.
So it should be protonated?
Yes.
Right. Now, does the protonated form look like this? Or does the protonated form have a positive charge?
It would be the first one.
Yeah, this one. That was a trap. We know this form doesn't exist because since there's no C in that side chain column, this is a acidic group. This has the potential to lose this proton. It doesn't have any potential to gain extra protons, so we don't need to worry about this. So the way you drew that was correctly. This is the correct protonated form. We don't want to fall into the trap of thinking there's another need for another proton. Now, is this pH low or high relative to this group?
That pH is high.
What's the consequence of that?
Deprotonation.
Okay. So that one, I think your first step is wrong. Now what went wrong over here, what might have gone wrong, what often goes wrong for me, is I forget who I'm checking. I might have said, oh, this is low compared to this. And that makes me think of protonation. So notice, you should not ask, is the pKa high or low? You should ask, is the pH high or low relative to the pKa? Ask, is the pH high or low? The pKa is just our threshold number. But they both have a p in them, so it's very easy to focus on the wrong number. Most of the time, it looks like you've been doing that right, but you can see how it's easy to get confused if that was the thing that gave you trouble there. We don't ask whether the pKa is high or low. We ask whether the solution has a lot of protons or not. If the solution has many protons, we would expect protons to end up on the functional groups here. How do we tell whether this is high or low? By comparing it to the pKa as a kind of threshold or a reference number. So relative to this, this was high deprotonated, low protonated. And he could also ask you what's the net charge on tyrosine here?
-1.
Because there's only one thing with the charge. All right, well, you should try to find some other practice problems like this and do some more practice with this. Amino acids is a big topic. We haven't even gotten into peptides yet. Remember that eventually we have to start putting these together, attaching one amino acid to another. All we've been doing so far is just looking at individual amino acids. So that's the bad news. There's a lot of stuff we haven't gotten to. The good news is I think we did make some good progress on some important stuff for amino acids. So we've seen how to draw the side chains, and we've seen how to start dealing with the fact that there's two different forms, for the main chain nitrogen, the main chain carboxy group, and sometimes for the side chain. That's why in the table, there's a column that gives you the pKa's for the main chain nitrogen, and there's a column that gives you the pKa's for the main chain carboxy group, and there's a column that tells you whether or not you need a pKa for the side chain. And just my experience is, even after I learned how to do these, I noticed that I tend to make lots of careless mistakes on these, maybe like you just made on this last problem. I definitely make lots of careless mistakes here. So when you're doing the test, you want to really try to make the time to double check your work on these problems, because even after you've learned how to do these, it's easy to lose the points that you deserve to get just with these careless mistakes. You want to try to do these problems twice, because like I said, even if you understand them, it's easy to make little careless mistakes like the mistake that we made over here. I make those mistakes all the time when I'm doing these types of problems.